Today, we'll talk about proteins. And we'll talk about the 4 structures of proteins for the IB. So, protein is actually polypeptide, which is basically a chain of amino acid. Okay? They all connect together with a peptide bond. But now, proteins you can differentiate it from primary, secondary, tertiary, and quaternary. So let's start with the first one. Now, for the primary structure, the key is that if you are able to tell me the number and sequence of the amino acid inside polypeptide, this is already the primary structure. This is actually determined by the base sequence that you transcribe and then you translate into a polypeptide. So the number and sequence of amino acids inside a polypeptide, this is what you call the primary structure. But now, if we extend that a bit more, we know that amino acid or proteins, they could fold into different shapes. So this will bring us on to the secondary structure. Now, if I were to draw a chain of polypeptide using different shapes, okay, I will now draw this as one line. This line can fold in 2 ways. The first way is in a helical shape, like a coil. You coil it up, we call this the alpha helix. Okay. But if you fold this up in a different format, okay, I will draw out these foundations first. This line could be like this. Okay, it's like folding like an origami. Okay, so this is called beta pleated sheets. Okay, both of this will fall under the secondary structure. Basically, it is, or they are, regular repeating structures from a folding of polypeptide maintained by hydrogen bonds. This is one of the key terms that you will need, okay? Because both of this would be maintained by hydrogen bonds inside the whole helical shape. The same applies here. The hydrogen bonding will occur in between. Because in your amino acid, actually you could have a lot of OH groups. They will give you hydrogen bonding. Okay, so we have covered the primary and the secondary. Now, tertiary is super easy because it's the 3-dimensional conformation or configuration of a polypeptide. But this is not just maintained by hydrogen bond. What do we mean by that? So imagine me joining this alpha helix to this beta structure right here. And they fold together again. Okay? In between, you could have many different attractions. You could have it here, you could also have it over there. There could be several possibilities. One, you could also get hydrogen bond. Okay, you guys better mark it down in case you guys forget it. Hydrogen bonding is one. You could get ionic bonds. You could get weak hydrophobic interactions, and the fourth one you could get covalent bonding. For the covalent bonding in the proteins, it will be focusing on the disulfide bridges. Okay, it's inside one particular amino acid. Okay, that will only form the covalent bond, and it will be a strong connection in between. All this bonding can form a further folding into a different shape to reach this structure. Okay, now you have learned the basics. We will now focus all this. This will give you one protein. If you have 2 or more polypeptide to make up one protein, what would that be? Okay, you will get a quaternary structure to form when 2 or more polypeptides form one protein. And those are the 4 structures of proteins. Thank you for watching, and remember to subscribe.